Okay, so let's come on to the next topic, that is Clover leaf model of transfer RNA. Correct? Clover leaf model of transfer RNA. Now we know very well, we have studied in 11th standard all the non-genetic types of RNA. There are two types of RNA, genetic RNA and non-genetic type of RNA, right? Genetic RNA generally constitute the genetic constitution in some of the viruses, but non-genetic RNA are the RNA which will play an important role in protein synthesis, right? So non-genetic type of RNA are of three types: messenger RNA, ribosomal RNA and transfer RNA. So, when you talk about messenger RNA, it plays an important role in copying the information, genetic information from the DNA and passes through the ribosome for the protein synthesis. Ribosomal RNA provides proper binding site for the messenger RNA to bind at ribosome. And transfer RNA will transfer the amino acid. So today we will start about transfer RNA. Transfer RNA is also called as soluble RNA or supernatant RNA or adapter RNA. Now, why this transfer RNA is generally called as soluble RNA? Transfer RNA is called as soluble RNA because this type of RNA, that is transfer RNA, is totally soluble in the cytoplasm and it cannot be separated by ultracentrifugation technique, right? Okay, so it is generally called as soluble RNA as it is totally soluble in the cytoplasm of the cell. Next, it is also called as supernatant or adapter RNA. Now, why it is called as adapter RNA? Because transfer RNA is generally having an ability that it can read the codon and simultaneously, right? It will read the codon and it will simultaneously go and bind with the amino acid, as the amino acid do not have an ability to specify the codon. So, as it goes and binds to the amino acid and it can read the codon, that's why transfer RNA is also called as adapter RNA, right? Okay, so transfer RNA is the smallest RNA of all the three non-genetic type of RNA, correct? It is about 15 to 20% of the total cellular RNA and the molecular weight is about 25,000 to 30,000 Dalton. That is the molecular weight of transfer RNA. Now, when you talk about this transfer RNA exists in two different forms. First is generally called as clover leaf like model of transfer RNA and second is generally called as hairpin transfer RNA, right? These are the two different forms of the existence of the transfer RNA. Let's talk about first talk about clover leaf model of a transfer RNA. Clover leaf or trifoliate model of transfer RNA generally consists of three major arms. You can see here first DHU arm or DHU loop, second T-C arm and third is middle arm or anticodon arm, right? DHU, dihydrouridinine arm or loop, is generally first loop which is meant for amino acyl binding loop. It means that amino acyl or the amino group will generally go and bind to the dihydrouridinine or DHU loop. Second is T-C arm or the T-C loop. It is the arm where the ribosome will come and bind. That is ribosomal binding loop. And the middle arm is also called as anticodon arm and it contains your anticodon. Anticodon of the transfer RNA will pair with the complementary codon of the messenger RNA to form codon-anticodon interaction, right? So how many arms? Three arms are present. DHU arm, also called as dihydrouridinine loop, right? T-C arm and middle arm. DHU for amino acyl binding loop, T-C arm for ribosomal binding loop, and middle arm or anticodon arm will contain your anticodons which will pair with the codons of the messenger RNA. So in between that you can also see a small a lump or a arm which is generally called variable arm or the variable loop. Okay, one is called as the long arm, second is called as the short arm. Long arm is generally having 3-N and the short arm is generally having 5-N, right? Okay, so this 3- long arm contains your CCA end, which is also called as amino acid acceptor end, whereas the 5- will be having a G end. That is 5- will be having the your nitrogen base G. Okay, G stands for guanine. So here you can see the long arm is having a CCA end, that is nothing but your amino acceptor end, amino acid acceptor end. Okay, so that's what all about the structure of a clover leaf model of a transfer RNA. Similar is with the hairpin like model of a transfer RNA, right? It is just coiled like a hair loop, right? You will be having 3' and 5', same CCA as the amino acid acceptor end. And last you will be having the anticodon that will pair with the codon of the messenger RNA to form a codon-anticodon pairing or the interaction. Okay, so now let's talk about the function of the transfer RNA. Transfer RNA, according to its name transfer, it transfers or places a appropriate amino acid at the CCA end, that is amino acid acceptor end, to the ribosome for protein synthesis. That is the major function of the transfer RNA. So this is the clover leaf model of a transfer RNA, also called as adapter RNA, because it is having an ability to read the codon and simultaneously bind with the amino acid, as the amino acid do not have an ability to read the codon. So it is called as adapter RNA, right? How many arms? 3 arms are present. DHU arm, T-RNA arm and anticodon arm. DHU for amino acyl binding loop, T-RNA for ribosomal binding loop, and middle arm or the anticodon arm will contain your anticodons, which will pair with the codons of the messenger RNA. Thank you.